Okay, thank you everyone. So welcome to today's webinar. I am Kai Leng, the Regional Field Application Scientist of Azure Biosystems. Today's topic will be on Fluorescent Western Blot 101, where how we utilize it to detecting phosphorylated protein. So before we start, let me introduce Azure Biosystems to you. We're a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. We manufacture the products that combine smart and simple workflows with high performance and affordability, so you can have the confidence in your data and move forward quickly. And also you have the flexible capabilities to go whenever the data takes you, whatever the research direction or positive or negative calling on that. So our company is based in United States, California, very near to Silicon Valley. Our company is founded by Arnold Shifji. He's our CEO and chairman. He's a venture investor, and also Dr. Diping Chair. Dr. Diping Chair is our co-founder and also executive vice president of R&D. If you're looking back his past experience or CV, you know that he has a lot of experience in developing protein imaging system back in Alpha Innotech and also Illumina. Here's some simple timeline on the company development. The company is a fairly new company, founded in 2013. All the core members are coming from Alpha Innotech. I think you'll be quite familiar with this brand if you've been doing chemiluminescent Western blot imaging. So they are one of the first um, manufacturer in the market that produce imaging system for chemiluminescent Western blot and also for some Western blot as well. Okay, so just a few key instruments in our company portfolio. We have Sapphire Biomolecular Imager, which is the industry first hybrid CCD imager and also laser scanning system. And we have Azure Imaging System, which is focusing on Western blot imaging, covering both Chemi and Fluorescence Western blot. And of course, we have qPCR machine as well. All in all, we Azure Biosystems have a range of gel system, reagents and consumables, and even imaging systems that for all your Western blotting needs, covering for both chemiluminescent fluorescent Western blot and for gel system part will be aqua gel tank. We even have like flash Western kit and so forth. So basically, if you need anything to do with your Western blotting, just if you have need, we have some solution for that. Okay, so now back to the presentation. In today's presentation, we have three important ideas to address. So the first thing will be Fluorescence Western blot meets the stringent publication requirement. And next, how this fluorescence Western blot is allow us to detect phosphorylated protein more efficiently. And lastly, how the multiplex fluorescent Western blot makes you validate the pathway easier. Okay, so we are basically utilizing one of the most important feature of Western blot, fluorescent Western blot, which is multiplexing capability. So first of course, I need to introduce the basics of fluorescent Western blotting to you. So just to give you a rough idea, why we talk about fluorescent Western blot as compared to chemiluminescence Western blotting. So fluorescent Western blot, as you can see, the keyword here is multiple signal on multiple proteins on one blot. Okay, so that's the idea for fluorescent Western blotting. How it done is made possibly by using two or more fluorescence dyes and an instrument that can excite and detect the light from each dye. Okay, so you can have two. Here we're talking about duplex here. Two primary antibodies can be probed at the same time for two separate proteins of interest, and then you can also probe the secondary antibodies at the same time, and that is conjugated to these for primary antibody and give you fluorescent signals. So we call it fluorophores. And these fluorophores, they have this very distinct, I'll say, feature called spectral separation, where they have distinctly apart excitation and emission wavelength one and another. Therefore, you can detect one protein apart from another. Therefore, the capability of doing multiplexing in a Western blot. Okay, so this is how it looks like on a multiplex fluorescence Western blot. This is a merge images of two channels. We have green channels and we have red channels over here. And of course, as compared to chemiluminescence Western blot, it's very basic, is one signal for one protein. Chemiluminescence itself, it is a reaction or chemical reaction generated light, then the light will be detected by either your CCD imaging system or an X-ray film. Okay, so the reaction comes by you having this enzyme either be HRP or alkaline phosphatase, and at the presence of luminol, it generates light as a side product. So therefore, it's a chemiluminescence, chemical reaction generated light. Okay, right. So when we compare the signal variation between chemiluminescence and fluorescence Western, and as you know, the enzymatic reactions, they are basically affected by a lot of external factors. For example, ambient temperature, the time of incubation, substrate concentration, pH, so and so forth. So therefore, it's been a widely known or accepted fact that chemiluminescence itself is not having or giving you that full range linear proportionality. What it means is the high protein abundance is supposed to give you high signal intensity and low and medium and so on and so forth. But the signal will change across time. It will also change when you use different kind of ECO substrate and so forth. So therefore, chemiluminescent signal is a dynamic rate of a reaction. It's a reaction. So as compared to fluorescent signals on the other hand is a static light of generation because there's no enzyme amplification of signal. Okay, so therefore it's stable. The only thing that can change the signal intensity will be the number of fluorophores on your membrane and also the light excitation, light, how say, input or the energy input from the imaging system. And as you know, the excitation light in the intensity from the system is a constant value. So therefore, you can safely assure that the is in signal intensity you get from your fluorescent Western blot is directly proportional to the protein amount that you want to detecting. And we have a lot more publications along the years since 2015 and so on and so forth, where they dive deep into comparing the chemiluminescence and fluorescence signals. And they come up with a conclusion that the fluorescent itself, they do offer more stable and linear light production because they are not an enzyme. And of course, not to say that the chemiluminescent signal itself are bad. No, if you look at this comparison between chemiluminescence and fluorescence signal, I'll say antibody detection in Western blot, they do serve differently to different needs. Chemiluminescence is super good in giving you the answer on qualitative question. Like, I just want to detect whether the protein is knocked down or is there or not. Okay, but it's not very helpful in giving you that relative quantification or even that accuracy and within sample to sample because it's been affected by a lot of external, I'll say, variables. But because this is an enzymatic amplification of signal, it is very, very sensitive. It can reach up to femtogram levels, and even people still using it today in a dark room to get a signals from very, very minute amount of, let's say, RNA, DNA, or even protein. Okay, it's a very familiar chemistry to all of us because I think that's the first, the widely used technique before the colorimetric Western blot. Okay, that will be a previous, I'll say the first version of how we visualize a Western blot. But now the commonly used will be chemiluminescence. And of course, it's compatible in both film, X-ray film, and also digital imaging system. But the different disadvantages for today is as we face increasingly amount of more stringent publication requirement and a lot of I'll say inconsistencies, okay, then we start to actually look back into the technique itself because all in all, you need to eliminate all these factors that said my protein is having low expression. Is it due to the technique itself or is it true that it is slowly expressed? So therefore, we have been a lot of people have been looking at this like how could we improve it, because we have been know that chemiluminescent signals is semi-quantitative at best, because the signal is depending on the enzyme kinetics, and it's very very good on a single protein detection. But if you want to detect another protein, let's say your housekeeping or so on and so forth, then you need to strip and reprobe the blot on a second target. And of course, if you're using X-ray film, it has a very, very limited dynamic range and saturation issue. So it's not suitable for common research or diagnostic use, because the expression is too high, and then it just saturate the X-ray film all around. Okay, and for chemiluminescence Western blot, it's very important that you need to resolve your proteins nicely, so they can separate nicely apart from each other. Therefore, you might actually cut the blot into several strips and do, incubation one target A on the strip, the top strip, or so on and so forth. Okay, so looking at this, even though chemiluminescence is super good in giving you that a signal for on low abundance protein, but nowadays, as I say, we are looking for something more, I'll say, efficient, more accurate, and even something that is giving us the reliable data. Therefore, Fluorescence Western blot comes into light. Okay, we have been having fluorescence dye for quite a long time, and then how we implement them on Western blot. So they do actually research on this and found out that the fluorescence Western blot itself, it can give you both qualitative and quantitative answers on all your questions, like whether there's protein or not, and then how much the relative protein expression compared to the other, and then and also the monitoring the entire Western blot technique, the QC of your Western blot. So in the context of where if you are not detecting low abundance protein, okay, but you need to do multiplex, okay, you need to utilize the multiplex capability to detect a lot of proteins at the same time, and then to avoid all the different blot to blot variation, blah blah blah, and humans errors and all these factors, then yes, fluorescence will give you more information in a single blot. It also increased quantitative accuracy because the signal by nature is more stable than the chemiluminescent signals, and it also allows you to archive and store the plot for a very, very long time, and you can re-image it later. And as a matter of fact, I and my colleague myself, we're still using fluorescence plot that we have like 3 years ago, and the signals are still very good. Okay, but of course, fluorescence plot themselves is compared to chemiluminescence plot, they do not how to say excel in part of the sensitivity. At most it will be picogram level as compared to chemiluminescence, which is femtogram level. And of course, when we trying to actually do fluorescence Western blot, you need to be careful about anything that will contribute to your background noises, like reagents, your membrane, and so forth. Okay, right. So there's a fundamental differences between chemiluminescence and fluorescence. And now we move to the part of why fluorescence Western blot allows you to publish data even faster or even more efficiently. Okay, we're looking at the Western blot publication requirement right now. Okay, just an example adopted from Journal of Biological Chemistry. You can see over here that actually not only about this journal, we have a lot of journals that's addressing this issue, and then because they do see inconsistency in data. Like, for example, if you have noticed this, if you change to a different lab, or if you've been doing the same research in a different country, you might actually see these inconsistencies. And then no matter how controlled you want it to be, and then we could not actually pinpoint what is the, how to say, the factors that are contributing to this. So one of the factors that people has been pinpoint will be the linear relationship between the signal intensity and the volume of your sample must be confirmed for every single antigen. Okay, so as I said, the signal intensity and also the sample volume, or I'll say the protein amount, it must be a linear relationship. Means how much the protein you're detecting and it should give you how high the signal it should be, and then it should be a linear relationship. Okay. And then for this, and under the context of without saturating the membrane. Okay, if you're talking about housekeeping, you also cannot saturate the membrane, putting too much of the sample. Then of course your control normalization will be very bad because it's not longer, how to say, valid. Okay, it's not controlled anymore. And then, of course, there's a lot more requirements in terms of Western blot publication. For example, they require you to publish, no, it's not publish, submit a whole blot image to show that your representative data or representative images or the images you put in the manuscript are indeed from the data, from the Western blot data. So to clear the doubts that they're having, scientific misconduct and so forth. And in terms of quantitation power, Fluorescence Western blot did excel chemiluminescence because it doesn't have all these variables that will affect the signal intensity. So as we know, the amount of chemiluminescent signals, they are subjected to a lot of variables like that will affect the reaction kinetic, like temperature, pH, okay, the concentration and the enzyme concentration, so and so forth. So if you want to continue to do your chemiluminescent Western, you have to make sure that you're using substrates with wide dynamic range and also have a longer half-life. Okay. And what to say by dynamic range is you need to make sure that your protein load does fit into the signal linearity over here. So you can see the line indicator over here. It represents the signal linearity or optimal signal range between okay, the higher and the lower end for that particular ECO. Each ECL have their own dynamic range, so you need to understand it and use it according to your scientific, I'll say, experimental design and so on and so forth. Okay, right. So for Fluorescence Western, you do not have that kind of concern because I said the only thing that you need to optimize will be your secondary antibody concentration. That's it. Okay, another thing about quantitative trade-off on chemiluminescence signal will be the membrane stripping and reprop problem, where you are having higher risk of losing your total protein, okay, when you strip and reprop for target B and target C. So most of the practice is we actually try to do, let's say, your target, okay, image for the first time on the blot, then you strip and reprop for, let's say, your housekeeping. That's one of the way of doing. In the context of post-translational modification research, like let's say studying phosphorylated protein and total protein, and we'll normally detect the phosphorylated protein first, strip, and then probe and detect your total protein. Okay, but as much as how we can control and how we can, reduce the amount of loss, there still be a risk or there is still be a loss in your total protein. So it's still very, it's not controlled enough. Okay, so if you've been noticing your post-translational modification research like between phosphorylated and total proteins are having inconsistency, then I will highly advise you to go to fluorescence Western blot straight away. So do not waste your time anymore. So all in all, for fulfill publication requirement, fluorescence Western blot is up for that because it preserves the whole blot image. When you image the blot, it will be capture the entire field of view. You do not need to cut the blot into several strips. And then do remember certain publication or journal, they request you to cut the plot into certain format if you need to cut them up. Okay, but you do not have to worry if you're doing fluorescent Western blot because every time you're taking the image, it will be entire blot image. And then you do not need to worry you might lost the data or because of a junior postgrad, they do not know how to capture the image or so on and so forth, because every single images that they capture will be a whole blot image. And of course, you'll be observing the loading control and project interest on the same image. So you're comparing bands on the same plot. Like for example, here you have phosphor, JNK, and the merged one, and also probably another your housekeeping protein as well. Therefore, there's no strip and reprop process, and there also no need to uh, if you've been doing total protein normalization, which is an even better control in terms of quantitative Western blot, then you don't need to prove your housekeeping protein that is linear to your signal. Okay, and therefore, fluorescent Western blot is improves your reproducibility and quantification or quantitation in a general in the long run and in immediately if you're facing these kind of inconsistency issues. Okay. Right, just some extra information on normalization and quantitative Western blot. Here, what I said about quantitative Western blot is Fluorescent Western blot, okay? Because it is quantitative, okay? Right, so for in Fluorescent Western blot, you can do housekeeping protein normalization. You can also do total protein normalization. The difference is very simple. You on housekeeping protein means you have one band that serve as a normalization control for the entire blot. For total protein, you're using the entire proteome as your normalization control for the Western blot. So the difference is very big here, one versus many. Therefore, the total protein would reduce your variabilities, your errors, and even experimental design, due to the biological variations a lot, okay? Because we are not looking at one particular marker, we are looking at a lot of them. So just some additional information on publication that how they're addressing the Western blotting issues. And one of the things that they're comparing will be total protein normalization versus, let's say, beta-actin or tubulin as your loading control. So you can see that this is one of the things that actually contribute to your reproducibility. Okay, so normalization do play the part in your final data analysis. Okay, there's some example. I'm showing you how it looks like on fluorescent Western blot. Housekeeping, you see 3 target protein plus your housekeeping, you can do that. And also total protein normalization and 3 targets at the same time. So this is a 4-plex Western blot, a fluorescent Western blot. Okay, so there's a maximum that you can see. In terms of quantitation power, not only about quantitation power, we're talking about availability or flexibility and efficiency to submit your public manuscript. Okay, the best will be fluorescence detection with total protein stain. Then next will be a fluorescence blot with housekeeping normalization, provided that you need to prove that the housekeeping expression is unaffected by your experimental design, treatment, or even variation in the biological sense. Okay, so of course, chemiluminescence Western blot also can do total protein normalization as well by utilizing a single fluorescent channel that actually giving you that better normalization control. So if you notice all these, I'll say better quantitation power or signal linearity, it will be more, we were looking at it at fluorescent signal side. We are not using it using chemiluminescence because like I said, chemiluminescence itself, it cannot differentiate different target for you. It just stain or it will just giving light in a single plot. Okay, so you can't really say, oh, this channel, this signal for target A, the channel another target, another signal for target B. You cannot do that. So the only way to achieve that is fluorescent Western blot and fluorescent signals. Okay, next let us move on to the important part of today's presentation will be phosphorylated proteins detection are more efficient with fluorescent Western blot. So just giving this is a case, also case study or a general a brainstorming session for you. So let's say you are detecting phosphorylated protein. You will need to show both non-phosphorylated and phosphorylated species. For example, I have a step 1 protein over here, which I did in my PhD study. So what I need to do is I need to I'll say analyze the protein expression of a phosphorylated step 1 protein and also a total step 1 protein. Therefore, we calculate, let's say, the amount of the expression of the phosphorylated protein, step 1 protein, related to the total. We are looking at the activated pathway activation, let's say. So this is unavoidable, and then that is very common, or usually there's no molecular weight differences between the total protein and the phosphorylated protein because they are basically the same protein, but one is phosphorylated, another one is not. Okay, so therefore, these two proteins are not be able to separate it by the means of gel electrophoresis. Because the molecular weight are so near to each other, you cannot separate them apart in the gel electrophoresis. So what happened is you have a slide, you have a blot, and let's say around 72 or 100 kilodalton, you have both step 1 and phosphorylated step 1 protein around the same location. Okay, so the only way to detect these two expressions is only strip and reprobe. I do phosphor step 1 detection, strip and reprobe the blot with total step 1 and capture the step 1 image. That's how we usually do. Okay. Right, so this are further as a worded form is if you're using chemiluminescence Western blotting technique, this can only be done by strip and reprobe process, i.e. probe with your phosphor step 1 antibody, image the blot, strip and reprobe with the step 1 total step 1 antibody, then image the blot. So this is how the data looks like. We do the phosphor step 1 first, then we image the step 1 photo step 1 later. Okay, so this is strip and reprobe process. So all in all, it takes longer time because now you need to, because as you know, when you probe for primary antibody, it will take another day. Okay, then you do your washing everything. So the entire, let's say, 2 days or 2 days Western blot will drag to 3 days or 4 days. Okay, and of course you require more reagent. Okay, you need to go through the entire process. Probing, blocking, washing, and so on and so forth. And then all in a longer run, the cost will be way higher. Okay. Right, so this can be easily overcome if you do it in the context of fluorescent Western blot. So how you do that is you use two separate fluorescent and secondary antibodies. So each bearing different fluorophores with non-overlapping spectra. For example, you have phosphor step 1, okay, that is conjugated, or you have a secondary antibody for, let's say, green channel 520. And then you have a total step 1 that is meant for the red channel, let's say 658. Okay, so there's a green channel, there's a red channel. So each responsible for that target. Then, as I said, when you actually probe them at the same time. Okay, okay. So what you can do is you probe these two proteins at the same time. Right, primary antibody, you're propping them together, incubate together on the blot, wash, then you prop your secondary antibodies together on the blot. Okay, then you carry on with your washing step, then you straight away go imaging. Okay, so there's some configuration on how to do that. So for example, your phosphor step 1 will be a rapid, then your secondary can be so this 800 channel, so anti-rapid 800, and for mouse step 1 will be anti-mouse 700. So this will be utilizing near infrared channels over here, which is 700 and 800 channels. Then this is the result that you get. Okay, without needing to strip and reprobe and risk losing your protein, you get the data, okay, on the same plot, okay, by image them at the two different channels. Okay, right. It improves your convenience and also accuracy because there's this reduced risk on uneven removal of the antibody and loss of target protein due to the strip and reprobe process, and reduce your Western blot protocol length to less than 3 hours. So this is under our kit called Flash, the Western kit that is a very quick one, and of course it gives you better signal linearity due to the fact that fluorescent signal itself is a static light generation. Okay, right. So that's why it is way better or more efficiently if you're doing phosphorylated protein detection with fluorescent Western blot. It saves you time, it saves you money, and you'll be assured and confident that the data that you get are not affected by, let's say, strip and reprobe process and so on and so forth. Because you're looking at one single blot and the blot having all the controls and all the groups that you have, and then you're comparing them within that blot. Okay, so and the signal is very linear, so there won't be any variations in terms of the signal linearity, and you sure that the signal that you get is true to that plot. Okay, right. So next thing, when we look at the phosphorylated protein detection more efficiently with fluorescent Western blot, we can extend it bigger. We are not only detecting two proteins right now, we are detecting more. Okay, so for example, in this publication, okay, is one of our users that published in the contact dermatitis. So it's impact factor 6.6 like that. And what they do is they're detecting toll-like receptor protein expression in fluorescent Western blot. Okay, so they have toll 1, 2, 3, 4, 5, 6, so 6 targets of them, and normalized with tubulin. Okay, then what they do is they use infrared, okay, conjugated secondary antibodies against rabbit and mouse primary antibody. So if I'm not mistaken, they're doing this for 3 sets, okay, triplex, okay. So it'll be 1 and 2 plus your tubulin, 3 and 4 tubulin, 5 and 6 tubulin. That's how they do, okay. And then do remember, even though multiplexing sounds very good, you do actually need to optimize the multiplex a bit. Okay, so nothing comes from free for free and nothing comes so easily. Okay, you still need to do a bit of optimization. It's the same thing like you do your chemiluminescent Western, a different setting, you still need to optimize it. Let's say you're changing the antibody a bit, then so on and so forth. Okay, right. Let us look at how the in the overview of the pathway validations. We're looking at toll-like receptor 1, 2, 3, 4, 5, 6. Okay, so they basically characterizing the entire toll-like receptor signaling pathway, and then the result they get is something like this. Okay, so you can look at the data, even though it's in grayscale color and also cropped out, you can actually present your fluorescence Western blot data in such a way, because the fluorescent data, it the visualization sense is easier for you to detect, okay, the expression changes by naked eyes when you turn it to grayscale. Okay, if you look at the data, so you can see by all these combinations of 6 toll-like receptor uh, protein expression normalized with tubulin, and all the grouping here, with the different, this is a different, I'll say the bacteria, and also a different skin and different condition of what kind of battle here, they come up with such a complex data that allows, that's all contributed by because of fluorescent Western blot. Imagine if you're doing this on chemiluminescent Western blot, it will take way longer time and way more money. And then, of course, if you're lucky, you may get the result nicely in a one shot or few shots. But if you're unlucky, then the optimization length will be even extended and taking a lot more time. Okay, let's look at another interesting publication by our research, our user as well, which is Science and Impact Factor 47.73. Okay, so this is part that we have. They are doing interleukin 33, phosphorus MET2, okay, and total SMAT2 and 3, and RF2, and of course they normalize with alpha tubulin. So they're utilizing the near infrared channels as well, IR 680 and 800 secondary antibodies. Okay, so I let me show. Okay, so this is a graphical abstract on what's the data or what kind of the I would say justification they're doing the research. Basically, they want to show that the tumor initiating cell, okay, TIC, they have this niche signaling loop to promote the cancer progression within the cells surrounding the microenvironment itself. Okay, right. So there's a result they show over here, okay, and over here, okay. So here the Western blot of lysate. So the knockdown is completely depletion of interleukin 33, and you have this one on looking at the knockdown and so forth to validate the entire pathway. Okay, so here's phosphorylation in control, phosphorylated proteins, and also the total protein plus interleukin 33, and also another one, a tubulin, that is for normalization, which they didn't show here. Okay, so this is how fluorescent Western blot 8 or help researchers to do pathway validation faster and easier. Because we are looking at everything in a single blot in a bigger field of view, okay? Everything in a single blot, so you don't need to worry about blot to blot variation, and also human error as well. Okay, so here, just a recap of what we are talking today. I'll be tell a little bit about how fluorescent Western blot meets the stringent publication requirement. Okay, and then with fluorescent Western blot, you can detect your phosphorylated protein more efficiently. It saves you time and cost. And if you want to do a pathway validation, for multiplex fluorescent Western blot even can help you in that sense, because you can do up to 4-plex, 3 targets plus housekeeping, or 3 targets plus total protein. Then you can actually characterize all the data and protein expression on the same plot. Okay, and then it's definitely saving you a lot of time and effort. Okay, so we are Azure Biosystems. We have imaging solutions for every lab and application. Of course, we have our own Western blot reagents that match for your Western blotting needs. And we have Azure imaging system, like I say, is focusing on chemiluminescence and fluorescence blot imaging. And also we have the Sapphire biomolecular imager. So just a little bit of product introduction. We have Azure Imaging System 600 model. I believe that, yes, Epical Scientific, they have a demo unit in their on the site. Okay, so if you're interested, you can always request a demo from them. So what happened in this unit is it can do both Chemi and Fluorescent Western, covering RGB and near infrared channels. So there are up to 5 channels over here, but for Fluorescent Western blot, we only recommend to do 4 channels or 4-plex, because if you can see, the 2 red channels are too near to each other, where the signal crosstalk is unavoidable. Okay, so if you notice that the two examples that I have mentioned before, a publication example, they've been utilizing the near infrared channels because the near infrared channels are using laser excitation light sources and giving you a very nice image with low or none background. And of course, the it's much easier to get the data. Okay, so because they no need to do match, so 2 is enough. Right, so there's some example on how people have been using this system for, of course, the standard DNA, the gel dot imaging. So it has all the UV lights and blue light and so on and so forth. Okay, ECL, okay, which is chemiluminescent signals with color marker, fluorescence blot, and in-gel fluorescence signals, and Comanche blue, and then we have the autofluorescence signals from the fossil, and bioluminescence bacteria. These are the mouse brain sections and mouse with RAP labeled subcutaneous tumor. These two are more for preliminary checking, and of course some markers from the plant and so on and so forth. And also fish, slides, and this is how our user has been using the machine for. And there's no limitation in terms of what you can do with the machine as long as the signal can be detected reliably. Okay, so some application on so this is actually produced by the Azure Imaging System 600. Okay, the science paper. And of course, we have the paper from Nature, Comanche, protein gel imaging, chemiluminescence, and of course, some other basic DNA gel imaging. I want to showcase the DNA dye detection limitation, and so forth. Yes, Western blot imaging comparison for this phosphorylated protein detection. Yes, this is actually available in the form of app notes. You can actually download this from our website to look at how the entire protocol has been running, and then how the data actually look for, and what are the reagents that they use for this experiment. And by imaging viral load in chicken embryos, just for the prelim study. And that's it. Thank you for your time here today with us. Okay, so I'm Kai-Ling, the Regional Field Application Scientist. We have Nicole with us today as well, and CK from our distributor, Epical Scientific, and also Jenny. Thank you. Also, I'll open the floor for Q&A sessions.